Okay, so good afternoon to this really exciting session, I think, but I'm biased. So my name is Dan Peer. I'm the head of the Precision Nanomedicine Laboratory at Tel Aviv University. And I was fortunate to do some of my work in Cesar Milstein lab back then in the 1990s. And then I moved to Boston, work in Tim Springer's lab, and we develop antibodies mostly for integrins. So, you know, it might be not the classical nano, but I think immuno oncology is becoming more and more interesting, and we have witnessed really amazing work and clinical translation. We already heard the talk of Carl June, and he should be here also in this session in a different talk. Our first speaker is Dr. Mark Kuzi from Janssen, whose talk is titled Bispecific Antibodies for T-cell Redirection Therapy. Thank you. These are short talks, and we'll try to be on time to make sure that we have some questions. Thank you.
Okay, thank you, Dan, for the opportunity to give this presentation. I come from near the Philadelphia area. As you probably saw also within Carl June's presentation, they have the motto, "The cure is within." And this theme is what I'm also going to share with you, this idea that there's a lot of aspects of what is available within the body that you can go about to direct what you want to have. The key fundamental concept that we work with lies in the concept of the antibodies, which have a particular domains. The elegance of antibodies lies in the large surface area that mediates this internal components together. By understanding that, you can actually control a lot of the aspects of how things can self-assemble so that you can direct what you want. So in one sense, this is the nanotechnology concept in terms of protein engineering. Now, one of the great themes that have come about is that if you focus on any one kind of a gene or protein-based system that's present within a tumor cell, often you don't get much efficacy because of the difficulties of understanding the nature of that one protein or gene. Because in many regards, if you look at the local environment that's going to be present around the tumor cell, the heterogeneous environment by which those cells live almost dictate that the responses that you would have would be very different. So despite the molecular understanding of individual proteins, a lot of the challenges involved with looking at tumors in a heterogeneous environment where they have the differences in gradients of both oxygen, nutrients, other kinds of regulatory cells, remains really a large challenge in terms of how you want to think about therapeutic interpretations. One of these strategies lies in being able to direct multiple mechanisms of action by which you can control this. So the ability to try to bring in as many components to try to control different kinds of pathways so that the synergistic effect can give you the efficacy and bypass the potential of resistance. This is what we're trying to do by using biologics, that you can control this potency, specificity, and the pharmacokinetic profile. So, traditionally antibodies, you have things which bind on to the target antigen, where you can have an antibody deliver drug conjugates. You also have the opportunity allowing the Fc of an antibody mediate a lots of cell-dependent toxicities, whether that's protein-based or cell-based. And of course, one of the themes that we want to bring about is whether one could elicit the opportunity of bringing in activated T cell so you would be able to bind to a particular tumor target cell environment. Now, there's a lot of interest in terms of how one wants to mediate such kinds of interactions. One can think about doing some wonderful protein engineering exercises because of the understanding about the individual components by which you can build multi-specificity. The great challenge, if you're going to be looking at any one distribution of how you put these things together, lies in terms of putting in components of red or blue specificities that are shown here. And you can dial in the desired potency specificity as you wish. But the big challenge has been, how can one make this routinely with lower cost of goods, that it's stable, that you know it is going to be compatible with the environment, so that you can understand the pharmacokinetic, pharmacodynamic properties, and making sure that if you're going to be thinking about doing this in a chronic dosing mode, that you will have less immunogenicity. So this has been the challenge. From that large number of constructs, which are numbered in a range of a few hundred right now, there is a much more limited subset that are undergoing and being employed in different clinical trials. And when we came into this field, we were concerned about the particular profiles which we mentioned, and what we decided to focus on is going beyond what's already launched in terms of applications of bispecific agents. There is a method of using antibodies, bispecific antibodies, to allow for targeting of a tumor cell for T-cell killing. There's another format that was mentioned in terms of single-chain fragment technology. But both of these suffer from the potential of being able to have immunogenicity profiles, PK profiles. So we decided to invest in looking at a more humanized bispecific technology, which we brought in collaborations with Genmab, which we have a variety of clinical trials being set up. In terms of this kind of technology, what this relies upon is being able to take any two antibodies of a defined specificity, introduce point mutations in the Fc part, the CH3 domains, and these designs of these mutations allow for these proteins, these antibodies, to be less stable as in the individual forms. And if you go through the process of reducing and oxidizing the hinge, you have a much more stable heterodimer formation, as shown by the crystal structures that we have described. By having these complementary mutations, you have a much more efficient heterodimer take place. So we wanted to employ these for T-cell redirection, the ability of having an adaptive immune response by adding these agents target particular tumor cells. Usually you think of T-cells linked towards a particular antigen specificity associated with a particular peptide, but we wanted to see whether we can reactivate these tumor cells, these T cells, in terms of acting on these cancer cells. And the concepts of having a bispecific agent which has a specificity for the T cells, allowing that to more or less bring about the activated T cells in the environment of an abnormal cell that has a particular target, so that by having that co-localization, you can have a formation of a T cell activation and turn on the destruction of the target cell. Now, it's critical when you do these kinds of experiments that you do not activate the T cells in the absence of the target cells. I mentioned the catumaximab, a particular antibody system that is a mouse hybrid. There is a single chain fusions which are being used right now to combat leukemia, was mentioned earlier. But what we wanted to more or less come about with is this intake of taking the hybrid or single chain fusions. Why not focus on having a full human bispecific antibody, less problems of immunogenicity, ability to control the Fc so that you have less potential depletion of the T cells, and use that as a way of doing T cell redirection. So by recruiting the T cells, we focus on CD123, which is overexpressed in AML. We want to use a bispecific antibody format, which allows us to have a longer half-life as compared to single-chain fusions. Having this as in a fully human form, you have the opportunity to avoid more antibody-drug antibody responses. We also introduce silencing in the Fc so that you have the potential of not having mediated killing of the T cells. And we have a variety of models which I'll allude to in a short while. So one of the first things we often do in discovery lies in more or less ascertaining that you have the cytotoxicity profile with different cell lines that have different levels of CD123. And you see only the bispecific species has activity. If you lack either the CD3 or the CD123 targeting arm, you don't see much of activity in terms of flatline profile, and you can reconfirm that in terms of having a cell line that lacks this antigen. We have done some xenograft models in which we look at having introduction of the tumor cells, introduction of our molecules of interest, and you monitor the size of the tumor that occurs over a period of time. And you can see that as you add more and more of this agent, you have that capability of controlling the xenograft growth control with the corresponding control profiles. Now, we focus primarily with antibodies, but we're also looking at the possibility of looking at different scaffolds. With the different scaffolds, you have the capability of putting on fusions onto antibodies with the appropriate kinds of linkers, and you can probe the questions whether architecture is going to be important for the potential of having target efficacy. Now, with regards to having a smaller sized molecule in terms of the scaffold, you also explore the wider range of epitopes that you can more or less direct these protein-based therapeutics. So for instance, if you take an antibody that has a recognition for a particular antigen, take this case of HER2, and you make another scaffold that has a recognition for another epitope within that protein. In this case, this is a afinimer targeting a different epitope than the already administered HER2 molecule. And you arrange the molecule, the afinimer, so it is at different particular locations within that antibody. What you can see is that you can, in fact, get different levels of efficacy in terms of killing of the tumor cells with the presence of the T cells. And this different efficacy is going to be related to the different kinds of geometry. This we don't really know in advance. This is something that we want to more or less engineer to see what the potentials may be. And this can also be a way of tailoring the kind of specificity for these kinds of engineering efforts. Alternatively, we can also take centurions, particularly against a particular target antigen, connect these onto an antibody that recognizes T cell antigen CD3 for a particular bispecific antibody where you have just, again, one arm against the T cell antigen, one arm against the T cell target cell antigen. You can modulate the specificity by just changing the location of the different centurion molecules which binds onto the tumor antigen. By the different kinds of architectures, you can see that you can in fact modulate both the efficacy and specificity in terms of that recognition. And this is now more or less part of the discovery process that we have over in Janssen in terms of trying to understand the best protein-based therapeutic to mediate this kind of a T-cell redirection effort. There's a lot of discussion that goes in terms of the engineering. If you take a very simple model of the synapse between a tumor cell and a T cell, you have a particular orientation of the components of the MHC presentation with the T cell receptor. There is a launch product associated with a particular single chain fragments that's linked towards blinatumomab. There is a particular distance in terms of how that recognition takes place. So if you imagine having a bispecific, there's a lot of discussions in terms of what's the optimal geometry in terms of what targeting could be mediated. Now, we don't know in advance what the best profile will be, so it is very empirical. And empirical in terms of how you're going to look at target density, which could affect the nature of avidity in terms of the engagement onto the cell surfaces. This aspect of exposure in terms of how much area, how much epitope that you really want to do, and trying to understand the threshold of what's the minimum amount of molecules to mediate that particular effect. These are all going to be dependent on the kinds of target cell antigens that you're going to work with, especially in terms of their signaling responses. And we've been doing the kinds of engineering in terms of the protein-based end of looking at the hinge in terms of how the binding arms attach with the linkers, being able to understand the nature of the silencing that you want to have. The half-life control, you can make it longer or shorter. It depends on how you do that kind of aspect of control, and an engagement of other kinds of agonism. So these are aspects that we are all working on over at Janssen. So in this short presentation, this is an effort that's ongoing between Janssen and Genmab. There's a lot of protein engineering as a way of being able to control redirection of cells. I just shared you some aspects of the T cell redirection, but we're also exploring other immune cell redirection, because it's never going to be quite clear in terms of whether any one particular modality of redirection is going to be successful. So with this, I close. I thank you for your attention, and I hope to answer any questions about the concepts we've presented.
So I'm curious about the flexibility of, so you have a hinge region. How flexible it is? Can you design linkers that...
Please use the microphone, we're recording.
Okay, okay, thanks. So how flexible is the hinge area?
Okay.
And also, how can you engineer spaces? If you know, for example, if you have a crystal structure of a protein that is on a surface of a T, of whatever you want, okay? How flexible is the bispecific antibody in terms of the length? Can you actually, you can measure it, at least from the crystal structure, and see if you have two targets that are in close proximity, how can you really engineer those, and how flexible is your hinge?
So with regards to flexibility, if you look at an antibody, it's my hands over here, the capability of having the spanning of about 10 to 20 angstroms is possible. Okay. Now, from doing biochemistry experiments, you can actually show that, in fact, you can have the Fab arm touch the FC in terms of biological assays. Now, in terms of that aspect of the control, you're always going to have to do an experimental evaluation. One of the aspects of antibody engineering is that there are different isotopes, as you allude to. And if you look at the difference between IgG1 versus IgG2, IgG1 just has 2 disulfides at the hinge area, it's highlighted over here, and an IgG2 has 4. Now, that provides a chemical constraint. So by doing EM or fluorescence or different measurements, you can in fact show that there is a difference in flexibility. And you actually see that as a manifestation and changes in activity. But we are not smart enough to know in advance. It's still very much an empirical exercise. Now, with regards to fusion proteins that are linked towards modulating specificities, you have the choice of linkers. It is a routine exercise that we will put a flexible linker, say like a series of glycine-serine combinations, and compare that to something that is stiffer, like an alanine-proline combination. Again, we are not smart enough. It's all very empirical. We often go through these iterations to see which provides you the best biological activity.
Right. Thanks.
Sure.
Thank you. I think that you almost answered part of what I wanted to ask you. So my question is, how relevant is that you use one method or the other? So when you want to have maybe to bring 2 cells together and that maybe one kills the other, it's very important if you have a kind of east-west conformation or north-south when you have the 2 specificities. You know what I mean? That you have a classic, because I think that, as you are mentioned, depends very much on the target and is very empiric, because the distance between the 2 cells probably is not much different with one or the other approach.
You're right. So, you know, when you talk about east-west, this aspect of east-west is that if you have 1-1, there's a different kind of interaction versus if you have 2-2. If you have 2, you have the potential of avidity, which could be useful for targeting things with low density. With low density, avidity could be your advantage. But you have to always check binding versus toxicity. If you have bivalent engagement of a receptor that turns on a response, that may not be favorable. So you can change architecture and geometry to try to allow for binding and not activation. So unfortunately, it's still very empirical. Why some targets work better than others in terms of monovalent or 2-2, that's still being explored right now. But it's a great question.
Have you tried in silico optimization by mutating in silico the amino acids? Did you try it?
So in silico, we're not that intelligent about this. In fact, we just use extensive libraries that are either put into an in vitro selection or an in vivo selection. So in silico, we are far, far, far away from knowing how best. You're correct in the sense that an antibody structure is very well known. We know exactly where the complementary determining regions are. And it's only a combination of 5 to 10 amino acids. But still, that range of diversity is still so broad, even when we see our hits, that we do not know in advance what the best combination would be until we do the experiments.
We are having some luck to do it with nanobodies.
So we've also do the same thing with nanobodies, and this concept of a different scaffold allowing for the engagement, that's also true. So we also use the camelid-based system in terms of looking for specificity and potency.
Thank you.
You're welcome.
Okay, thank you so much.